Receptor clustering is ubiquitous and often necessary for the signaling cascade activation. However, few methods are available to measure the degree of clustering. We use Total Internal Reflection Fluorescence TIRF microscopy to follow the diffusion of FGFR1 mEGFP molecules in the plasma membrane of live cells. Then, we apply number brightness, the N&amp analysis, to describe the dynamics of stimulated receptor clustering. TIRF is ideal for fast temporal imaging of molecular events that occur near the cell surface, while N&amp determines the average oligomeric state of fluorescent molecules based on their diffusion in and out the illumination volume of the microscope. Indeed, this is a tool to connect the spatial-temporal organization of the receptors at the cell surface with their signaling. N&amp only needs the access to a microscope with a fast acquisition module. You can analyze large area of live cells, having just a basic knowledge of fluorescence fluctuation methods.
This method can be applied to proteins that form dimers or clusters and can be stoichiometrically labeled with a fluorescent dye. If the proteins are in intercellular compartments, other types of microscopes are used for acquiring the images. It is important to prepare a construct that expresses the monomeric form of the fluorophore for measuring under experimental conditions the pure monomeric brightness as a control.
On the first day, seed HeLa cells in 1.5 milliliters of complete medium at a concentration of 100,000 to 200,000 cells per milliliter.